Today I am presenting the functions and secretion of bile. Of the lecture is as below: physiologic anatomy of the hepatobiliary system, composition of bile, role of gallbladder, functions of bile, regulation of bile, these three phases: cephalic, gastric, and intestinal phase. Then gallstone formation. These are the headings in which I am covering this topic. Now you see here the diagram taken from partly from Genon and partly from Guyton. Here is the liver. You have the liver there, and this liver have this hepatic duct system, and this hepatic duct system comes through here and enters the duodenum here. The common bile duct, this is a bile duct, common bile duct joins with the pancreatic duct, makes the that becomes the common duct. So now if you are coming here, the same thing is here. This is the liver, these duct systems, the liver duct systems, and you have a gallbladder here. And this gallbladder, okay, and the bile duct comes here and joins with the pancreatic duct, makes a common bile duct, or hepatic and pancreatic duct is a common, empties into the duodenum through the ampulla of Vato. This is a brief about the anatomy of these things. It's a typical hexagonal thing, and you have those triads situated at each place. This is a triad of a portal vein, hepatic artery, and bile duct. Lymphatic duct, so whatever the things reabsorbed, they will enter through the thoracic duct, they empty into the atrium. So that means all things reach the heart. So that is all the metabolic activities, synthesis, the protein, fat, and carbohydrate synthesis, or synthesis of other molecules, are emptied into these sinusoids. Whereas certain secretions from these cells, bile is a secretory product, and the bile contains excretory products. So that means excretory products, especially the hemoglobin metabolism or other detoxifying molecules, or the molecules which are conjugated, the steroid hormones which are conjugated and they are excreted here, so that they can be cleared through the urine or eliminated through the urine. So like that, this contains bile acids and the other products which are to be excreted into the intestine. Now, these bile duct, you see that this bile duct now enters into the duodenum. This comes here, enters into the duodenum, and empties it as required, because it's not continuously being emptying. So it empties only when the sphincter relaxes, or because of the contraction of the gallbladder and the ductal system, and that empties here. That is one aspect. Having considered this aspect, we go further. What is bile? Bile is a secretion from the liver. That's what I mentioned. It has the secretory part, that is bile acids, and it has an excretory component, that is bile pigments, other detoxifying or detoxified molecules, and it also contains electrolytes, and the pH of the bile is alkaline. That means above 7, and the composition resembles nearly the pancreatic juice. Nearly 500 ml of bile is secreted every day in healthy individuals, and some of the components of the bile are reabsorbed in the intestine and then excreted again by the liver. So that means we have an enterohepatic circulation. So that means especially the bile acids which enter into the intestine and they come back, they are recycled. So this is enterohepatic circulation. The bilirubin is a breakdown product of the hemoglobin. You know, the hemoglobin, mainly the spleen and other endothelial systems, they break down, and once the hemoglobin is breakdown of the bilirubin. It is an insoluble form, so that bilirubin bound to the plasma protein albumin reaches the liver, and once it reaches the liver, this gets conjugated, and this is known as conjugated to form bilirubin glucuronate or bilirubin sulfate. That is the soluble form, and this soluble form is excreted as through the bile duct. Suppose if it is not glucurinated, if it is not conjugated, so then it will remain attached with the albumin. So that is another thing. What happens in case of hemolytic anemias, in which there is a large amount of bilirubin is formed. This is not conjugated, and there is a enzyme system for conjugation of these bilirubin are limited in the liver. So there will be a increased unconjugated bilirubin. So that I will not go into detail here. The soluble form is excreted in the bile, as a bile pigments, bilirubin, bilirubirin, and these give a wonderful golden yellow color to the bile. Okay, so the composition of the bile, you just see that 97% of the bile is water. Salts, it contains bile salts, they are about 7.7%. Bile pigments, that is a hemoglobin end product, it is about 0.2%. And cholesterol, it contains 0.06%. Inorganic salts like bicarbonate, sodium, potassium chloride, and all those things are 0.7%. And we have fatty acids 0.15%, and phosphatidylcholine is 0.2%, and it contains a fat about 0.1%. It also secretes this alkaline phosphatase, and it is excreted, and this alkaline phosphatase is also there in a normal value. Now, during resting stage, what happens? The sphincter is not open. If this sphincter is not open, whatever the secretion from the liver is not emptied here. If it is not emptied here, then what happens? The pressure builds up in this duct system. That means the secretion keeps here. So then it gets back into a back pressure that enters into the sac. This is the gallbladder. This is a pouch there, and this enters into this pouch, and this pouch is the storehouse of the bile, because during a resting state, this bile should not be wasted. It should not enter here unnecessarily for doing nothing. So this is stored here, and that is what during resting phase, a sphincter is contracted. A sphincter OD is contracted. Do not allow the flow of bile to duodenum. It does not allow this component. And if that is not allowing, the biophysical forces, that means they will divert this bile to enter in this pouch, that is gallbladder. And the gallbladder is not a simple pouch, these things. It has the epithelium, gallbladder epithelium, concentrates the bile by 5 to 10 times. 5 to 10 times. And then it will get emptied as required. So that is the function of the gallbladder. So that means it holds the bile at a resting period and concentrates it and empties it as required. The concentration may be 5 to 10 times, sometimes maybe we will see that what is that. So this is the composition of the liver bile and gallbladder bile. Gallbladder bile, so water is decreased, water content is decreased, so that means it is concentrated. Bile salts, they are increased, so 6 gram. So that means it's about 1 gram here, it is 6 gram. So bilirubin is increased, 0.04, 0.3, 7 to 8 times almost. Cholesterol, 0.1 gram, it becomes 0.3 to 0.9. So it is concentrated by 3-fold. The fatty acids, So this is 0.12 here, and it is 0.3 concentrated by 2 to 10 times, 2 to 10 times, depending upon the secretion. Then the lecithin is 0.04, and it is again 8 times. The sodium 145, and it is decreasing. It is decreased sodium level because the reabsorption of water is reabsorbed, water reabsorption along with the sodium. The potassium increased, calcium increased, chloride, it's 100 milliequivalents here, it's 25 milliequivalents, it's decreased 4 times. Bicarbonate, it's 28 milliequivalents here, and it is decreased. That means there is a 2.5 times, or it become 1/4, 1/4 or 2/5 of this thing, or 2/3 of the concentration decreased, 28 to 10. So that means in the gallbladder bile, specifically what we have seen, there will be in concentration of all constituents excepting sodium, chloride, and bicarbonate. Sodium, chloride, and bicarbonate. All these things are not are decreased. Now, going further, the secretion of a bile, how does it take place? It takes place from the hepatocyte, and it has two mechanisms. So this synthesis of bile is by two mechanisms. One, bile acid dependent mechanism, bile acid independent mechanism. And bile acid dependent mechanism, if I take it further here in this hepatocyte, this is the hepatocyte, the liver cell. In this hepatocyte, we have made, this is a bile acid dependent component, and this is a bile acid independent component. The first part is the sodium potassium ATPase. So that's the beginning of this thing, keeping this cell active or in the form of the polarized state. The sodium and the potassium, that the sodium potassium ATPase, this works, and this is the first part. And because of the sodium potassium ATPase, there is a gradient of sodium. And this gradient of sodium, say, look here, the sodium enters here, and this is the co-transporter, sodium bile salt co-transporter. This this imports bile salts. So the bile salts are there, bile salts move in along with this transporter, and you require, this is sodium. The sodium enters in, and because the sodium is less here inside, and the sodium is more outside, the sodium enters in, and the sodium entering in along with the bile salts. And these bile salts now are conjugated with taurine and glycine, mostly glycine. And these conjugated bile salts, as a glucuronate or something, they will be again transported. These are again another transporter, bile salt transporter here in the lateral spaces. So they secrete this thing, the lateral space. Along with that, the cholesterol, phospholipid, and the bilirubin, that is the hemoglobin product, which is conjugated bilirubin with a large amount of bicarbonate and sodium. They are there. So now this bicarbonate and sodium is coming from this side. So this is a bile acid dependent because of the presence of the bile acid in the jugular or canaliculi, and that would stimulate this thing to enter. This stimulation of this thing will activate this because the sodium potassium ATP is already there, and that transport these components. Then if you are looking at here, so since it's a metabolically active tissue, because carbon dioxide is produced, and this carbon dioxide, carbonic anhydrase catalyzes or has a catalytic, forms the carbonic acid. Carbonic acid is dissociates into bicarbonate and H ion. Now the bicarbonate ions are pumped into the lateral space, and the H ions are pumped with the sodium H ion exchanger pump. This is sodium exchanger pump, and the H ion is taken out, and this is sodium comes in place. And now this sodium again enters here because HCO3 is entering, sodium enters here. That makes the entry of HCO3. And all these molecules exerts osmotic activity. And this osmotic activity drags water, and this water moves it through the canaliculi further. This is what I have explained here. So I have just indicated 1, 2, 3, 4, 5. 1 is sodium potassium metaphase, and the 2 is a bile salt sodium synthol transporter, bile salt transporter. 3 is a carrier mediated transport. So that means you have another carrier for the conjugated bile salt to be transported, and then the fourth and fifth are bile acid independent flow, and that is with the sodium potassium exchanger. This is sodium potassium exchanger there, and bicarbonate transporter. And this bicarbonate transporter is associated with the chloride, because for one bicarbonate transported, the exchange, it exchanges one chloride. So that means it is a bicarbonate chloride exchanger type, that type of a transporter. So this is what happens, how the bile is secreted into the canaliculi. Now what happens in the canaliculi? These flow in two, because the canaliculi, in this canaliculi, they have added up, and once they are added up, they should move it further. And the movement of these bile secretion will depend upon two factors. One, the factors in the duct, and another factors in the canaliculi. In the canaliculi, we have a bile acid dependent mechanism wherein bile salts are secreted into the lateral space, into the canaliculi. And bile acid, this one is a bile salts are in this bile acid dependent flow. The bile salts are being added up into the system, and that would increase. And bile acid independent, it's only bicarbonate and sodium, and this is happening with the bicarbonate and sodium. The flow, as the bile acid secretion increases, the flow increases. This flow increase is mainly the bile acid dependent flow. Bile acid dependent flow will increase, and this remains constant all the time. It remains constant because it cannot have. Similarly, the ductular secretion, it is because of the more and more bile acid. See, it is parallel with the bile acid dependent flow. The ductular, the total bile flow is parallel with the bile acid dependent flow. So you just see that once it is increasing, this is also increasing. This is the total bile flow. So this is how the bile flows from the system into the duct system. And now this, what is this bile? The precursor, these are the bile molecule of the bile. The cholic acid and the chenodeoxycholic acid are the bile salts, are the bile acids. These are acids, and now these acids do not remain, so they remain as a conjugate or a deoxygluconate or taurate. So in that form, cholic acid or cholic taurate or cholic gluconate, something like that, they exist. So now these are primary bile salts. So these are originating from this sterol ring. The precursor molecule for the bile acid is a steroid nucleus. The steroid nucleus either comes from cholesterol, vitamin D, or steroid hormones, or digitalis. These are the sources for the precursor molecule. Now the bile acids are secreted into the bile, are conjugated to form conjugated with glycine and taurine amino acids, and this forms cholic acid gluconate or cholic acid taurate. So cholic acid, primary bile salts are primary means they are synthesized by the liver, so they are not being corrupted. So they are either in the form of a cholic acid or chino or kino deoxycholic acid. These are a primary bile salts. You can just see that the number of percentage of these salts, it is 50%. The cholic acid is 50%. The deoxycholic acid is 30%. So that means nearly 80% of these bile acids are this nature. Or practically all of them, are the remaining part are secondary because they form in the intestine and in the terminal part of the intestinal segment. They get by the bacterial action, they get converted into deoxycholic acid. It's the lithocholic acid in the colon, or ursodeoxycholic acid. So these are the secondary bile acids. These secondary bile acids because of the bacterial action from the intestine. So these two are a secondary one. Deoxycholic acid is 15%, lithocholic acid is 5%. So that means lithocholic acid and so they are lesser. So this is total composites of bile acids in our body. Now, what this bile do? So now you have here a situation in which this is a pancreatic lipase, an enzyme secreted from the pancreas, and now it is digested into triglyceride. And if there is no bile salt, there is no bile acid, so now all of them would be in this form. All the fatty acids or the monoglycerides, triglycerides, and all the fatty products will be in this form. Whereas in the presence of bile salts, what is happening? These would form what are clusters. Clusters are grouping. These are called micelles. So the micelle formation, this is simple micelle formation, and this micelle formation getting emulsified and reaching it. This is the enterocyte. It is the intestine. It's an intestine, and they reach to this site for the absorption. So that means the fatty acids are liberated by the action of pancreatic lipase. Okay, and the bile salts make them in the micelle form, these circular structures. And the hydrophilic and hydrophobic domains, they attach the fat molecules. So these polar peptide bond, they will put them together, and then after putting them together, their arrangement will be in the sort of a circular domain. It becomes like a cylindrical structure. It will be like a cylindrical structure, as we have seen here. This is a cylindrical structure. They will be in the cylinder as a cylinders arranged beautifully. So these all the fat molecules. They hold on to this hydrophobic together, and hydrophilic is inside. So this type of arrangement the bile salts do. So that means by doing so, they form the micelles. That means a globular structures, cylindrical discs. They are having a number of fatty acids held them together. So that is one function, the formation of the micelles, because of the empipathic nature of the bile. So bile as such is like a soap. It's like it has a detergent action. By the detergent action, it reduces the surface tension, especially it is secreted in the first part of the duodenum. And once it is secreted in the first part of the duodenum, so that will try to, because the pancreatic lipase is added up, and there is a lipid component, and it will reduce the surface tension of the lipid component. Then allows the spread of the lipid molecule. and the spread of the lipid molecule and the agitation of the duodenum, the movement of the food or a chyme in the duodenum in intestine, because there is a to and fro movement of the intestine happening, because some part of the part is entering from the pyloric emptying and enters into the curvature of the duodenum, and once it is coming, and there will be small amount of back and forth movement, so that would produce a sort of agitation, and that will emulsify. The surface tension is lowered, so that fat will be spreading, and then because of the movement or agitation of the intestine, and along with other molecules like phospholipids and monoglycerides, with the help of these segmental movements, they will form the emulsification. That means it's an emulsified thing, just like your milk. Milk is an emulsified solution. So then bile salts added to this thing. So in addition, the bile salts are added to this. That would be having both hydrophilic and hydrophobic characters. They hold the hydrophobic in one clusters, forming the cylinders, what are called micelles. And after forming the micelles, they ferry or transport these micelles to the brush border. They transport these micelles to the brush border. So that is the next function. So if you are looking at here, so this is formed here in this thing, and this is the intestinal brush border. So they are transported here to the brush border, and once they reach the brush border, they will get attached to the enterocytes. So that means they will get attached. So then once they get attached, that will be absorbed. Either it is in the free form, that will split them into single fatty acid, or it will be cell drinking process. Cell drinking is known as a pinocytosis. It's a form of endocytosis that would engulf the entire molecule, and that enters into the cell, and that is released into the blood circulation. So that is how it aids the reabsorption of the fat. So that means the preparation, surface tension, emulsification, then micelle formation, the ferrying, then pinocytosis. These are the processes the bile salts perform. In addition, bile salts, what happens? The bile salts stimulate their own production. That is the bile salt dependent secretion in the liver. So the presence of bile salts, what happens? The bile salts enter into the intestine. They enter the portal system, and then they are in the portal vein, and they reach the liver, and then they will stimulate the liver cells to secrete bile. So that means they are recycled. So that is the cholaretic action. That is the recycling action of the bile salts. So that means not all the bile salts which are secreted or excreted in the gastrointestinal system, nearly 90% are recycled. Only 10% will be excreted. So this is about the bile salts. But thinking about, I'm talking about up till now, I'm talking about the functions of the bile salts. Functions of the bile is more than the function of the bile salt. So bile salt is one of the constituent of the bile. The other thing is it has a large quantity of bicarbonate. If you see that the bicarbonate level is high. So now the bicarbonate level is high, so that it will neutralize the acid chyme which is coming from the stomach, so that it helps the pancreatic bicarbonate secretion, and these things together, they will try to neutralize the acid chyme appearing in the first part of the appearing in the duodenum. So in addition, the bile performs the excretory functions. What are those excretory elements? It eliminates the bile pigments. It eliminates or excretes the bile pigments. Especially, I want to recall here, this is the bile pigment which are in a soluble form. The bile pigments which are insoluble, as it happens in case of a unconjugated bilirubin. Unconjugated bilirubin, it gets attached with albumin and remains with the albumin only. So this conjugated bilirubin appears in the bile, and this is eliminated. Now, once it is eliminated, this would be taken in the colon as a urobilinogen and stercobilinogen that gives the color of the urine and the stool. Then the bile salts, bile itself stimulate the production of the enzymes of the pancreas, because bile alone cannot do. Bile has a limitation. If you are looking at the bile, bile has no enzyme system, nothing to break down. Whereas pancreatic lipase is there in the pancreas, and that pancreatic lipase is required. So for that reason, the bile presence of bile or even the bile salts will activate the pancreatic the SNR cells to secrete the pancreatic lipase. These are the functions of the bile. What is a steatorrhea? A steatorrhea is a condition in which there is a excess amount of fat in the stool. Excess amount of fat in the stool. What happens without the bile salts? If there is no bile salt cup, that means if there is a stone in the common bile duct, so then what happens? The bile will not appear in the duodenum. So that means it will, without bile salts, up to 40% of the ingested fat are lost. So that means you can see that 40% of the fat we ingest is lost. And it will be lost in the feces. You have a very bulky and pale stools are formed, and this bulky pale stool, which is known as a steatorrhea. Steatorrhea. So this is a condition in which there is the deficiency of bile salts. It may happen in case of a stone in the common bile duct, or the tumor of the head of the pancreas, or even because of the infection of the liver, hepatitis. All those things may produce a steatorrhea. Because of the steatorrhea, number of other things also follow through, and these, especially the fat and the fats, number of metabolic deficits because of the fat and the fat soluble vitamins may result. So this is a brief note on the steatorrhea. Now, what is how the bile secretion is regulated? I have mentioned it is being regulated by nervous and humoral factors. So I have put the nervous in a nervous stimulation is coming from the vagus. It is the small component, only 10% of the component. More than 90% of the component is by the humoral factors. And these nervous and humoral factors, as we have seen with the pancreatic secretion, it follows the cephalic phase, gastric phase, and intestinal phase, this format. This cephalic phase and gastric phase, they include, especially cephalic phase, include those conditioned and unconditioned reflexes. The sound, sight, smell, and taste of food, or even the mastication, the esophageal movements, gastric movements, they will also influence the secretion of bile from the liver. Okay, but these two constitute only small fraction or small proportion of the bile secretion. More than 90% of the bile secretion is by the intestinal phase. That too, here I am telling you, in the intestinal space, the first part of the duodenum secretes a secretin. The secretin increases the HCO3 secretion from the hepatocytes. HCO3 secretion, that is the bile acid independent mechanism. Bile acid independent mechanism. Then we have the CCK, the cholecystokinin. Cholecystokinin, actually it contracts the gallbladder and it opens up the sphincter body. So that means it evacuates or it empties the gallbladder. Okay, so now this emptying of the gallbladder takes place, and that is by the cholecystokinin. At the same time, it will stimulate the hepatocyte activity to secrete the bile ducts. Say, for example, in case a person who is undergoing the cholecystectomy. Cholecystectomy is a surgical procedure in which the gallbladder is removed. Gallbladder is removed from an individual because it has a stone or a disease or something. Now, you may ask a question. If you remove the gallbladder, then what happens to his nutrition? Gallbladder is a storehouse of the bile. Suppose if it is not there, so this duct system may accommodate some sub quantities. This person then second question comes to you. If gallbladder is removed, then what happens to the person, whether he will be able to perform the fat digestion and absorption properly? Yes, certainly he will be able to do it, but now the duct system become more efficient, and the CCK activates the duct system, because the duct systems are also having those myoepithelial cells which are sensitive to the CCK. So now gallbladder is an organ, maybe even its absence also does not make much difference to our life. Now bile salts, they will stimulate actually in the intestinal phase the recirculation of the bile salts, what is called enterohepatic circulation, that enhances the bile secretion. So then gastrin, because the gastrin is secreted here in the stomach, so now that would stimulate the hepatocytes so that it releases the bile, because it will tell that okay, this becomes a part of the gastric phase. The kinin and other amines, because they only try to empty the contents of the ducts and the gallbladder. So here you can just see that vagal stimulation causes a weak contraction of the gallbladder. That I have already said, this contributes very little. Bile acids via blood stimulates the parenchymal secretion. Bile acids, the enterohepatic circulation. Secretin via bloodstream stimulates the liver duct system. So that means here it stimulates the liver duct system, and it will also activate the bile acid independent mechanism. Bile stored and concentration up to 15 times in the gallbladder. Now, the cholecystokinin via the bloodstream causes the gallbladder contraction and relaxation of the sphincter body. Then only it empties. With this broad outline of the regulation, we go with the enterohepatic circulation. The enterohepatic circulation is a very important concept for your examination point of view. Here I have taken it from Rhodes and Flanger. So now this is the gallbladder there. This is the duct system. This is the liver, and these are the conjugated primary salts, bile salts. They enter here in this duct system and stored in the gallbladder, and they are released as required in the small intestine. So as the bile salts appear in the intestine, they are being reabsorbed. They are being reabsorbed into the circulation. This is the portal circulation. This arrow, big arrow, is the portal circulation. So these are reabsorbed. The number one indicates the passive reabsorption. This is a very small quantity. Such type of the bile salt reabsorption passively contributes only 5 to 10%. The carrier mediated transport, because there are carriers in the intestine which transports these bile acids back into the circulation. They are large number. This happens 80% of the cases. 80%, more than 80%. It's only 5 to 10%. So then these bile salts are split into deconjugated. So that means a conjugated bile salt with a glucuronic acid or the glycine or a taurine, and they will be deconjugated, released, and these free bile salts enter in here. So this one, nearly 90% of this thing comes back. 80% here, 5% here, and even less than 5% here. They all come here to the liver. That means entero, this is intestine, hepatic circulation of the bile salts. So that means it comes here, it stimulates the bile acid dependent secretion up from the liver, and it comes here again, it is released, and it goes on, and such type of thing is happening. So the whatever the amount present in our total storehouse of the bile salt is not all emptied. Only 5 to 10% of that thing is excreted as a deoxycholic acid or lithocholic acid as secondary bile salts in the colon. It is about 500 milligram, that is 5 gram, 0.5 gram. Total bile acid pool is about 6 gram. You can just see that. So that means it's only 10% of that thing is lost daily in the stool. The remaining is recycled. So this is an important concept. You want to remember enterohepatic circulation. I repeat here, the bile acids secreted here are reabsorbed from whatever the mechanisms, 1 to 3 mechanisms I mentioned. One mechanism is the carrier mediated transport, the passive diffusion, and the deconjugation. So they all enter into the portal system, they reach the liver, stimulate, they then come back into the bile duct, and this type of recirculation happens, and that only small 10% or less than 10% is excreted. This is enterohepatic circulation. Then a concept about the gallstone formation. Bile salts are formed in the hepatic cells. You already know. These bile salts, while they are forming, nearly 1 to 2 gram of cholesterol is also being secreted in the bile each day as a product, because the hepatic cells from cholesterol, because it is coming from the cholesterol, and along with the bile salts, the cholesterol is also secreted. Cholesterol is insoluble in water, so it's a fatty thing. It is insoluble in water. The bile salts and lecithin, they keep the cholesterol in the form of ultra microscopic micelles. The bile salts and the lecithin, they just keep them in a micelle form, the cholesterol. Okay, so now such thing happens because in the bile, the cholesterol is there, the ultra micelles of the cholesterol present, and that is in the colloidal form. When the bile becomes concentrated in the gallbladder, because in the gallbladder it is concentrated maybe 10 to 15 times, the bile acids and the lecithin become concentrated along with the cholesterol. Everything gets concentrated, which keeps the cholesterol in solution. Because of the proportionate concentration of all the products, it does not. Under abnormal conditions, what are those abnormal conditions? The obesity. So that means cholesterol levels are high. And when the cholesterol levels are high, so that these cholesterols cannot be maintained in this form of a solution or in solution, that means the cholesterol gets formed into a microcrystals. Or when we consume a high fat diet, continuously we are consuming a high fat diet, so that will also enhance. Or the inflammation of the gallbladder. In the inflammation of the gallbladder, then it increases the water reabsorption and the epithelial damage. So these two conditions increase the concentration further, and all these things precipitate the formation of cholesterol gallstones. So these formation of microcrystals of cholesterol on the surface of the already inflamed mucosa, and that acts as a nucleus, and that grows, and gallstones are formed. And these gallstones may occupy, there will be number of gallstones in each gallbladder in this sac, and they may obstruct the the duct of the gallbladder, and that is a choledochal, the gallbladder duct, and that may produce pain and other abnormalities. For that reason, the stone is the most frequent culprit for a gallbladder removal. Because gallbladder, it may not jeopardize your fat digestion, and even there will be an adaptability of the duct system to hold on to the whatever the bile in the duct system. Now move on. So same thing, I mentioned about too much absorption of water from bile, and too much absorption of bile acids and from bile, or too much cholesterol in the bile, or inflammation of the epithelium, all produce the gallstones. These are gallstones there, and these gallstones may come here and stick in this duct, cystic duct, and that may produce pain and other abnormalities, and it may lead to an emergency situation. And for that reason, you remove this thing and this grows. So this is about the formation of the gallstone. Now, to end, today I had discussed about the bile formation. It's in the liver, the bile acid dependent mechanisms, bile acid independent mechanism, various functions of the bile as a whole, because it has both the excretory as well as secretory functions. The bile salts, they have a special functions of reducing as a detergent function, or forming a emulsification, forming a micelle formation, the pairing of the micelles, absorption, pinocytosis, and even stimulating its own production. The excretory function is eliminating the bilirubin, the soluble bilirubin, and the detoxified agents. And finally, it will be a stimulus for the intestinal and pancreatic secretion. These are the functions of the bile, and I mentioned about the regulatory aspect of the bile. I mentioned about the enterohepatic circulation. So these are the references I have referred: Guyton's textbook of medical physiology, Genong's review, and Bernard Levy. So this is, and today I have referred the Rhodes and Flanger, another textbook, some one figure I had taken. Okay, next class I will briefly cover the motor functions of the gastrointestinal tract. In these motor functions, I will cover the swallowing and defecation reflex in little detailed manner. Okay, now you can try to work on these some of those assignments. I'm trying to describe the composition and functions of bile, add a note on regulation of the secretion of bile. You can prepare. Then gallstone formation, the functions of bile, the micelle formation, steatorrhea, hepatic bile and gallbladder bile, and enterohepatic circulation. Okay. Thank you.